Machine learning for plant species classification. Using leaf vein morphometric. Abstract: the number of plant species are extremely huge, with about 391,000 vascular plant species all over the world. Hence, it is impossible and not practical for a botanist or an expert to be able to identify and classify all the species. In addition, some plant species may have high similarity between each other, taking a long time to differentiate them. Hence, there is a need to develop an automated or computerized system to identify and classify plants. With the advancement of science and technology, machine learning has been widely employed for classification and recognition tasks in many domains, especially in the biological fields. Automated plant species identification system could help botanists and laymen in identifying plant species rapidly. Machine learning is robust for feature extraction as it is superior in providing deeper information of images. Plant species identification is a research field area, which includes pre-processing, segmentation, feature extraction and classification. In this field, there exists many algorithms to analyze the image. The paper aims at understanding pre-processing, extracting the leaf features using contours and classifying using various machine learning techniques, namely, support vector machine, SVM, Nearest-Neighbour, KNN, and random forest, RF. It presents the leaf of various plant species from which the vein characteristics extracted, presented to detect and classify various kinds of plant species and other artificial intelligent techniques used to perform pattern recognition. The features extracted using contours found to fit well with the RF classifier, which is little greater than SVM and KNN classifier respectively. Keywords: leaf vein, segmentation, feature extraction, classifier algorithms. Introduction. Plant species classification is one of the interesting fields in which machine learning techniques are applied, to differentiate between the species. Automation in plant species classification is done using extracting features of the plants. Leaf shape is the most commonly used feature, that is used to develop such automated plant classification systems. Other than shape, leaf features like color, texture, and veins can also provide additional information that may be help in automation process. With the advancement of science and technology, machine learning has been widely employed for classification and recognition tasks in many domains, especially in the biological fields. Machine learning techniques, such as the support vector machines, Nearest-Neighbour, and others are artificial intelligent techniques are employed in pattern recognition. In this work, contours is applied to extract the features from images of selected tree species. The dataset used for this project is the Flavia dataset. The data pre-processing technique used here is data transformation. The numerical dataset has obtained as a transformation of image dataset. The extracted features that are in the CSV file fed into few classification approaches for learning and training purposes. Classifiers that had employed in this work are support vector machine, SVM, random forest, RF, and Nearest-Neighbour, KNN. A conventional method Sobel edge detection was replaced with contours, as the latter is memory efficient by removing redundant points. 2 related work. The number of plant species are extremely huge, with about 391,000 vascular plant species all over the world. 1. Currently, machine learning, a subfield of artificial intelligence, AI, is a popular and widely used technique that has been applied in various domains including biology, medical, computer vision, speech recognition and others. 2.5. Deep learning is a modern AI approach, which contributes a robust framework towards supervised learning. 6. It is able to map an input vector rapidly and efficiently to an output vector even in a large dataset. 7. Image enhancement is a process that is used to emphasize the features of an image. 8. Texture is one of the important features of the plant identification system, which can be used to characterize the leaves based on the surface structure of the leaves. It is a non-consistent spatial distribution pattern of different image intensities 9,10, which concentrates mainly on each single pixel of an image. COPE ETAL 11 introduced an evolved vein classifier based on genetic algorithms, GA, and ant colony algorithms to extract the vein structure. ANIMI ETAL 12 had proposed a combination of color and texture features based plant identification system. LARES ETAL 13 constructed an automated leaf identification approach for legumes based on the vein architecture. Only simple measurements were applied on the vein morphology and then identified by a random forests approach. CATIR ETAL 14 proposed another method on the foliage dataset and the Flavia dataset. LI ETAL 15, 17 proposed a CNN technique to identify 44 plant species acquired from the Royal Botanic Gardens of Kew, England. The extracted features were then classified with a multilayer perceptron, MLP, and a SVM. Two different datasets were used, namely, whole image, D1, and leaf patches, D2. Both datasets achieved an accuracy of more than 97%. Furthermore, researchers had combined both local and global features together in 18 and achieved more than 91% accuracy. Furthermore, Slotojevic et al. 19 employed CNN for plant diseases recognition. Another study based on leaf vein morphological patterns and using deep learning technique for plant identification was proposed in Greenblatt et al. 20. 3 proposed work. The classification of plant species is considered vitally important in preserving biosphere and helps botanists to identify huge plant varieties without more human effort. The data pre-processing technique known as data transformation is used to transform the plant image data set into numerical data set with necessary features as attributes using contours method. Further, classification is done using 3 machine learning algorithms, namely, support vector machine, SVM, random forest, RF, Nearest-Neighbour, KNN, and the accuracy of these algorithms are compared and the algorithm that gives best accuracy is used for further testing of real-time images. Fig.1 shows the workflow of the proposed work. Fig 1, workflow diagram of proposed plant species classification using leaf veins. 4 methodology. The proposed work has 4 main steps as shown in Fig 2, which are collecting dataset, image pre-processing, feature extraction and classification. First, the leaf samples were collected and images were acquired from Flavia dataset. The leaf images were then pre-processed and fed into the feature extraction step to retrieve the important information from the leaves using contours approach. Lastly, the extracted features were trained and classified by using various machine learning methods. Fig 3, images of 32 different species present in Flavia dataset. Here we show 1 image per species. B. Data pre-processing. Data pre-processing is essential in machine learning as data we use may be subjected to noises, which may affect or suppress the quality of the data. Noises occur as pixel values, which do not represent the true intensities of an image during the image acquisition. It is a necessary step to remove the image noises in order to highlight or enhance the important features of an image. The leaf images were reconstructed into square dimension, MXM, which was required as the inputs of data pre-processing. The original images with 6016 x 4016 resolution were resized into 1600 x 1200 resolution in order to maintain the ratio of the leaf shape. The images were then retained in the RGB format. This background removed image is used for further processing. Fig 4 shows the pre-processed image and segmented images. Data is transformed from image format .jpg to numerical data that is well organized and improves. It can be utilized for both grouping and relapse errands. In data quality and protects applications from potential context, it can be inferred the appropriateness and landmines such as null values, unexpected duplicates, usability of random forest calculation and a few other vital incorrect indexing and incompatible format. Things about it. The flow of execution is shown in Fig.5. See feature extraction. Features play an important role in differentiating the different hand signs. However, the selection of feature requires the proper understanding and interpretation of extracted feature values. First, all images were converted from RGB images into grayscale images. Then, contour was used to segment out the region of interest, ROI, from the images. After segmentation, the images were then post-processed and skeletonized to ensure a clean image of the leaf obtained. The vein features were extracted from the segmented images by measuring the vein morphological features. The leaf area was computed for the shape, color, and vein texture, endpoints, entropy, aspect ratio, areoles, inverse difference moments, rectangularity and so on. Declassification. Classification, as the last step for an automated plant recognition system, is an intelligent algorithm in training data to recognize the specific features of each individual plant species and categorizing a new sample as the correct species. The favored machine learning methods for plant identification are artificial neural network, and support vector machines, SVM, and Nearest-Neighbour, KNN. 3 classification methods that was used in this work were SVM, KNN, and random forest, RF. Support vector machines, SVM, a supervised machine learning approach, is conceded as one of the powerful classification methods due to its high capability in dealing with high-dimensional space and data points that are not linearly separated. Applying linear SVM on feature map data can execute speedy with low storage and improve the classification performance. Linear SVM with one versus all scheme was employed in this research since it involves a multi-classes dataset. A KNN is a classification approach, which classifies a sample according to the majority vote of its neighbors. The number of neighbors in this research is fixed as 1 with the city block distance metric. Random forest is an adaptable, simple to utilize machine learning calculation that produces, even without hyperparameter tuning, an awesome outcome more often than not. It is additionally a standout amongst the most utilized calculations, since its effortlessness and the way that results and discussion. From the context of this work here we have discussed the performance of the different models. After the data pre-processing, for performance evaluation the data was partitioned into training and testing set in the ratio of 70:30 and fitted to 3 different classifiers, and then we extended our work with testing with some real-time images of the plant species. The performance of each classifier is discussed in Table I. S.NO classifiers accuracy. 1. Random forest 90. 2. Support vector machine 88. 3. K nearest neighbors 84. Table I. Classification accuracy of different classifiers. It is observed that random forest classifier achieved the best performance of 90% for accuracy. However, the other classifiers obtained accuracy of not less than 85%. Hence, in this research we can claim that RF is highly suitable with contours for feature extraction model. In contrast, SVM and KNN were less suitable with the contours. On the other hand, conventional feature extraction method extracts each type of feature separately and manually, consuming a lot of time. For example, if the shape features are considered in this work, different sets of processes will be required for segmentation followed by shape feature extraction. Thus, machine learning approach is more practical and appropriate than conventional methods for developing an automated plant species classification system. 6. Conclusion. We proposed a shape-based approach for leaf vein morphometric with several steps including sampling, image pre-processing, edge detection, feature extraction, classification, comparison, etc. The strength of this approach includes its simplicity, time management, accuracy, ease of implementation, and it does not require any significant amount of training or post-processing. It provides us with the higher recognition rate with minimum computation time. The weakness of this method is that we define certain parameters and threshold values experimentally, since it does not follow any systematic approach for vein recognition, and maximum parameters taken in this approach are based on assumption made after testing number of images.